on a sec. There we go. Now we've got the recording on. Hi, my name's Amy. I am a data scientist at Zindi. I'm calling in from South Africa. I've had the incredible privilege of working with a team from around the world. Just to name a few of our sponsors and the team I've been working with, I've had people from Phillips, from Stanford Medicine, from SEMA, University of Navarra, Fudana University, Leibniz, University of Hannover, and most recently the Microbiome team. It's been an intimidating yet inspiring learning curve, and I think I might be sold as a bioinformaticist now. So this is our first webinar of 2 webinars. So this is the introduction to the MPEG-G microbiome classification challenge, where your task is to predict where a microbiome sample comes from on the body based solely on metagenomic data. That's a technical term, which we'll get into a bit later, so don't worry about that. I'm just going to change the view to speaker view. Brilliant. I'm going to share my screen and I'm going to hand over to... what happened here? Jan, you are going to have to share your screen, please.
No.
My screen's having a bit of an issue.
1 second.
Thank you.
There we are. Sharing the PowerPoint. And if I click on play from start, you should
Brilliant.
see the slides. Okay, perfect. Okay, so I'm Jan from Leibniz University, Hannover, here in Germany, calling in from Germany here. And we've been working for quite some time on an international standard for representation of genomic information. So it is my pleasure today to introduce you here to the MPEG-G standard very briefly, and to our open source software Genie that is used to store and transmit the genomic data in the scope of this challenge here. So let's dive right in. So here's a slide on the motivation for this international standard. So the story goes as follows. You see here the DNA sequencing costs per human genome in US dollar. And this is a figure going from the year 2001 up until recently. This is a couple of years ago already, but the numbers did not change much. And you see it's a logarithmic scale. So the sequencing of a single human genome, of the first human genome, this was a multi-billion-dollar effort by a large consortium. And then you see that the sequencing costs dropped quite drastically in the recent, let's say, 2 decades. And right now we have sequencing costs which are a few $100s per human genome. And what this led to is an inflation of the DNA sequencing data that is being generated and stored and transmitted. And to give you some ballpark numbers, when we have a typical clinical study involving, let's say, a few hundred or 1,000 patients, we already have to have a storage infrastructure with multiple petabytes of storage. So it's really a problem to store all of these data nowadays. So there were multiple universities, entities, companies coming together, constructing a standard for this. So here you see again the example I just showed. So when you have a single human genome, it's 1 to 3 terabytes per individual that is generated. And now we have this standard that is called MPEG-G. The last G stands for genomics. It's a standard series with the goal of producing the means to store and transmit this genomic data. The goal is here that existing de facto standards, maybe you know some of them already, like FASTQ, SAM, BAM, VCF, CRAM, which are there and which are widely used and which can also be used. But the problem with these standards is they are not maintained, and there's no organizational backing behind them. So that's why these multiple entities and companies, industry, they saw the need for an international standard. Let me quickly introduce to you what this MPEG and MPEG-G and everything stands for. So the story behind this is there are international standardization organizations. Most prominently, this is the International Standardization Organization, ISO. And there are also other organizations such as the International Electrotechnical Committee, IEC. And IEC and ISO, they have a joint technical committee, and this committee standardizes a lot of things. One of their groups is called MPEG, which stands for Moving Picture Experts Group, because historically this group was constructing all these video and audio codecs. You might already know. For example, the MPEG group developed the MP3 standard, which is an acronym for MPEG-1 Part 3. That is the audio standard practically everybody on the entire world used for hearing music a couple of years ago. Now there are more modern standards such as AAC, and a similar story goes for video coding. There was AVC, HEVC. And now this group has also made a standard for genomic data. A standard is nothing more than a PDF file that you can download from the internet. So a standard needs to be implemented. And that is what we did in a quite large consortium. But before I come to our implementation of the standard, let me quickly clarify some of the terms you might see along the way when working with the data. So MPEG-G is our way of saying this is the international standard series ISO IEC 23092. And here on these slides, which can also be shared or will also be recorded here in this webinar, I will not read through it. You will see here some explanations of some terms, and you can refer back to them whenever you might need to. So here you see who developed the standard, that it's facilitating data verification, etc. Let me now come to the structure of the MPEG-G standard, and you see it consists of many, many parts. It's a very complex standard series. For your reference, we will be looking at part 2 of the standard. For us, it's important how we store the genomic information and how we can efficiently transmit it. So the other parts are also are basically there to support part 2 of the standard, the coding of genomic information. Now, as I said, we have implemented this standard. It's a pretty boring PDF file, lots of text, but you can actually implement this. We did this. If you're interested in that, it's the language C++. It's available on GitHub, and it's also published in a journal. And this implementation of the standard, we call it Genie, which stands for Genomic Information Encoding. So, and this is, by all that we know, the prominent open source implementation of MPEG-G that is available on the internet and in general. I will now show you a very complicated image of what happens inside the Genie software. You at all don't need to remember this. It's just to make clear that this is like a very complicated process, and a lot of thought has been going into this software and how to organize the data and how to transmit and code it efficiently. Here at the bottom, I have put two let's say, shell commands. And this is maybe the most important thing here for you that you need to know. It's how to compress and decompress data in the MPEG-G format. And also in the starter notebook, you will see kind of these commands again. Let me quickly walk through them so that you can see what it's doing. Let me quickly switch on the pointer. So what you see here is we are using a Docker image from Docker Hub. And we run our tool, we specify which working directory we want to mount to the Docker container. Then we run the software with the latest tag from Docker Hub. We give it an input file. This is just DNA sequencing data. We give it an output file. And this is our compressed output file, which is very small and can be nicely transmitted over the internet. And then when we want to decode the data again, we again call our Docker container, we put it to work, and now as input file, we give it the compressed data, and as output, we get the genomic data again. So this is what it all translates to. It's an enormous MPEG-G standard. It's a nice open source implementation. It's good to know some of the basics behind it, but in the end, you will most likely be fine by using just these or like similar commands in this challenge. Last but not least, I have to say this MPEG-G and Genie activity, and this MPEG-G activity foremost, is like a collaborative effort of many individuals. I've put an incomplete list of names just here and of organizations. All of these people have been involved in designing the MPEG-G standard. Not all of these have been involved in doing the Genie software. The Genie software has been done by the people that are listed on the repository or on our publication. That would be a very quick journey from my side through the topics of MPEG-G and Genie, and I'm open to take any questions if you have some. Thank you.
Amazing, Jan. Thank you so much for that. I'm busy chatting away. I just wanted to say thank you so much, Jan. That was a great presentation. I'm going to hand over to Nevenka now to tell us why did we even decide to do this challenge? Where did we get this data? Why was this study even done? And then she's going to go into a bit about what are microbiomes. What is 16S rRNA? That's something I didn't know 2 months ago. So over to you, Nevenka.
Thank you, Amy. I'm just trying to find my way around all these windows.
Relatable.
Okay. Got it. Okay. So I can share my screen. Okay, brilliant. I learned brilliant from Amy and many other things. So anyway, I'm very excited to present at this webinar. And thank you, Amy, for all the collaboration. And Jan, thank you for a brilliant introduction to MPEG-G, MPEG genomics. My own professional life started with MPEG because my PhD was actually in extracting video features and analyzing video using MPEG encoded data. So how do we go from compressed bits to meaning? So that's why I'm so excited about this hackathon. And without further ado, let me put my slides in a slideshow mode. And I want to say we prepared this with my colleague Xin Zhao, now from Fudan University in China, and formerly he was at Stanford University. Xin could not be here today, but he's the first author on the study from which we get the data for this hackathon. So let me explain at sort of 30,000 feet elevation. So this is really an exploration at the intersection of AI, microbiome science, and health. Here we work with real-world multi-omic data, and that's why this study is so exciting, because it's not only microbiome data, but also there is representation of metabolomic data, as well as immune-related data like cytokines, as well as clinical events. So here we learn how genomics data can be compressed and analyzed, as Jan explained, and then modeled along with immune profiling using modern machine learning techniques. And I believe that you all are geniuses at machine learning, and also you'll step up to this amazing challenge of learning more about biology, especially related to microbiome and health. And then we collaborate with experts across the world. I definitely would say, if you know a biologist or a medical doctor who's your friend or a colleague, bring them in and collaborate, because that's how you will learn, and that's how you can make your own code, your own models much more applicable to the real world. And of course, we want to develop tools and insights that advance integrative and scalable bioinformatics. Now let's start with what is microbiome. All the years when I was growing up, bacteria was a synonym for something really bad. We were not really thinking about commensal bacteria that actually does something good in your body. And in the recent years, this has changed a lot, especially by studying microbiome. There is international human microbiome effort, multiple studies, many people across the world. Myself, I have worked on detecting oral cancer using saliva sample, meaning how do we study the microbiome and connect certain species to the presence of certain species to as a biomarker for oral cancer. And that's being developed now as a clinical test. Now, it's really a community of microorganisms. So bacteria, viruses, fungi, archaea that are living in a particular environment. Now, when it comes to humans, key microbiome sites are gut, meaning stool samples, skin, oral cavity, meaning saliva samples, nasal passages, as well as urogenital tract. And as we learn more, we learn that there are many, many important functions of this ecosystem that's between the human genes and all the microorganism genes in our body. And this is related to immune modulation, nutrient synthesis, pathogen defense, and many functions in the body. And when there is imbalance or dysbiosis, then this is linked to diseases like gastrointestinal diseases like irritable bowel disease, obesity, cancer, neurodegeneration. And every day you will see in the scientific literature many new papers regarding some connection between the microbiome and some disease. Even things like depression is linked to the status of the microbiome. So having said this, as Jan mentioned, in 2001, the first human genome was sequenced. It was about $3 billion. But now you can sequence a human genome only for a few $100. and you can sequence microbiome sample also for about that price, depending on the depth of sequencing that you want. So current research definitely suggests that the number of microbial cells in the human body is roughly comparable to the number of human cells. Previously, we thought this is a 10-to-1 ratio, and that's why the picture on the right side, I've included also the article. It's really interesting to read the full article. It's written only a few years ago. Many parts are still holding. And the estimate for humans is that in our body, we have about 30 trillion cells and about 38 trillion bacterial cells. So while the number is very big, the total mass is only around 7 ounces. So that's why this figure is on the right, like the proportion of the weight of the microbiome. And the microbiome catalogs today have about 200,000 genomes. When we started microbiome research a decade or more ago, this was like 10,000 genomes, meaning these are reference genomes that are assembled from multiple species. Then we went to strains, which is very specific strain of a species. And it's very important to note that human microbiome also includes other organisms and that bacteria make up the largest proportion. And one of the most well-studied microbiome areas is really the gut microbiome through stool samples. And we can get information about different bacteria genera, species, and strains. In addition to human microbiome, people have studied soil microbiome, river microbiomes, even New York City subway microbiome, microbiomes in airplanes, and so on, many, many interesting things. So embarking on this challenge also gives you an opportunity to acquire skills for studying microbiomes in many different environments. So let's focus now on the study at hand, the data that we have, and Amy will walk you through the notebook next. But first, on a very high level, this is a very interesting study because it consists of data that goes over 6 years from 86 participants, and this is across the gut, skin, nose, and mouth samples. And what we've learned is that there is persistent bacteria that are unique to each person, and this is shaped by genetics, diet, and immunity. It's like a biological fingerprint. This is a link to the study, and of course, you're welcome to read this in depth. You will need to understand what is the analysis done to date so that you can aim for more novel analysis. And what was studied also here is people with type 2 diabetes. And as we know, type 2 diabetes is the new epidemic in the world as we have access to a lot of food. And as people today, quote unquote, suffer with obesity, and some people even call it diabesity because they frequently go hand in hand together. And this is why it's very interesting to study the role of the microbiome in type 2 diabetes. So while this is over 6 years, many people are sampled once per 3 months, but some people are sampled very often, or there are samples when they go through episodes of antibiotics, different types of infections, and so on. So in total, there is over 5,400 samples. And as they summarized, around 118 million data points. One of the key findings, there are many, many key findings, but the fact is that long-term multi-site monitoring reveals that the microbiome is a fluid, systemic ecosystem, means it changes. If you change your diet, your gut microbiome will change. Excuse me. So I don't know why I lost my voice, but I'll put myself on mute so I can have a drink of water. Sorry about that. Here we go. And this is the overall study design. At the top left, you can see that every 3 months, there is sampling of individuals. So the sample collection goes across blood in order to derive all of this fascinating data. The full proteome, so this is from plasma, also a metabolomics profile, 724, and proteome. These are untargeted. Untargeted means you will get any type of proteins, but you're not specifically targeting for specific proteins. As opposed to cytokines, there are 62 cytokines, and we know exactly in that Luminex assay that we are getting those 62 cytokines. And also there are lab tests. For example, C-reactive protein, which indicates some kind of an infection in the body, and many others. And it is fascinating to have such an in-depth and personalized following of all these people over the 6 years. And then from stool, skin, oral cavity, and nasal, we have the microbiome samples and the microbiome sequences, which are encoded in MPEG-G data, which will be decoded using Genie, as Jan explained. Here on the B panel, there is the sample number and all the different types of measurements, the lipidome, the skin microbiome, oral microbiome, etc. at different time points. So this is also a very high-level overview. And very interestingly for you, because this is part of our first challenge, is to come up with, in an unsupervised way or self-supervised way, to show how these samples, all of these 5,000 samples, how do they cluster into similarity by body site, like oral samples versus stool samples. And as you can see from this figure here on the UMAP analysis, the skin and nasal, there is some kind of a not very specific boundary between skin and nasal, because nasal is definitely also part of skin, partly skin, but it's kind of a protected cavity. And this is when you asked in your questions, how is this different? Is this novel? So in order to arrive at this picture, there is a standard bioinformatics analysis of aligning the reads the genomic data from microbiome against catalogs of microorganisms, counting, and then we come up with, as you will read in the paper, with a clustering method for these samples. Now, the challenge here is to start with not that catalog and the standard bioinformatics analysis, but to start with the reads, to go directly from reads all the way to this kind of a super high-level representation of the classification of the samples into body sites. And also, I think the next two panels are just showing the richness and the evenness of the presence of different microorganisms across these body sites, as well as how the prevalence varies and the general rank of the presence of these microorganisms. So let's dive into the data. I'm sure you have downloaded, some of you have already started to build their models, but let's go through this. So we have 5,432 microbiome samples, which are taken quarterly as well as during these events like infection, vaccination, antibiotics, so before and after the event, so we can see the deviation from normal. Also that this is in 4 body sites: stool, meaning from the human gut, then oral from the mouth, nasal from the nose, and skin samples. And the analysis here data. We will talk more about this next time. And we have here longitudinal data of circulating cytokine levels. Now, what is cytokine? Those are signaling molecules that are part of our the work of our immune system. And the analysis is really the mixed effects models. This is figure 4 in the paper, used to identify systematic correlations between microbiome taxa and cytokine levels. So the interplay between microorganisms and our own immune system, as well as health and clinical data. So I invite you to explore and also discuss and ask questions on the forum. But it's important that this contains events, many events that are recorded, meaning time of infection episodes, vaccinations, antibiotic treatments, as well as data collected so we can link these physiological changes with the microbial dynamics. And also the focus here has been insulin resistance versus insulin sensitivity in type 2 diabetes. And this is where microbiome stability and diversity is perturbed. So can you get a microbiome sample and predict whether someone has insulin resistance or insulin sensitivity? That's another interesting challenge. So as Jan introduced the sequencing data and how we're going to work with sequencing data, I just want to go on a super high level to explain what this means. 16S is the ribosomal RNA gene, which is conserved in bacteria, but with what we call hypervariable regions, meaning they're variable across different species. It really enables identification of bacterial taxa in these complex samples. And there are steps, which is first we perform, I think this is meant RNA extraction, PCR of the 16S gene, then sequencing, denoising, and clustering and taxonomic classification. And then the output, which is what we all as data scientists are interested in, is OTU versus ASV. I will explain this in a second. Diversity indices and phylogenetic trees. And just so that we have a perspective, what is 16S versus other types of microbiome sequencing. So there are three types in general. 16S, which is our data that we start with from the Snyder study. So it's focused on bacterial and archaeal identification using ribosomal RNA. It's very low cost, less data, and it's taxonomic only, meaning you can have taxonomic assignment, but you probably don't know what the activity is. Then there is shotgun metagenomics. So all of the DNA is sequenced, not just a small part, meaning we can analyze bacteria, fungi, and viruses. And this is, so we can infer functions and taxonomic profiles, and it's higher resolution than 16S, but it's more complex. And recently, Viome joined our challenge, and we will talk about their data at the next webinar on end of July, where metatranscriptomics, this is RNA. So these two methods, so here we only look at DNA. If DNA exists, doesn't mean that it's expressed. And this is why metatranscriptomics is so important, because we know all of the RNA transcripts, and we know the active, meaning the functional and the taxonomic profiles. And this is the highest resolution, but it's most complex. So a pipeline can run a couple of days to analyze a microbiome, a set of microbiome samples, even in modern architectures. So now you say, okay, we will perform various types of deep learning and machine learning analysis. What we are going to learn from this? So I think there is always this note of caution and distinction between correlation and causation. Most likely, we are going to be in the correlation and association space. So here, it's easy to design. Causation is much harder, but wink, wink. This is where we are hinting at. Although in biology, nothing is considered causative until you can perturb one gene at a time or one parameter at a time and really show the causation, say, in an animal model, like in a mouse model. And that's hard to design and hard to prove. And techniques really are in observational study, like this one that we are looking at. It's case control study. So for each person, the normal state where they feel healthy would be really the case, the control. And then when they fall sick and so on, then that would be the case. And the advantage here is that this is large scale, it's hypothesis generating. And this is really the point, that maybe we can generate in this hackathon new hypotheses. And this is why this is so important and motivational for all of us to be part of this challenge. However, we can pose hypotheses that will be disproven at some point, which is also okay, because that's how we learn. And then examples, there are many microbiome projects. So in addition to the data in this study, you are also welcome to dive into some other data on your own. And the typical findings are the correlation between, say, microbiome composition, so some kind of an imbalance, some kind of dysbiosis with a certain phenotype. What do we call phenotype? For example, someone with type 2 diabetes, that's what we call diabetes phenotype. Or say someone has taken antibiotics, the state of the gut microbiome after taking antibiotics would be considered a different phenotype. So this is a very high level description of the technology which is applied in microbiome research. So sample is taken, and first the 16S rRNA gene is targeted. Then there is a step of PCR amplification, which then goes through high throughput sequencing. And from this point onward, this is where the data comes in. This is where MPEG-G comes in. So the data is stored, and the second row is all analysis. So cluster similar sequences, we derive abundance matrix and derive identity taxonomy. There are many existing tools which we are not going to talk about today. But you're welcome also to look them up. But the point is, how do we go from raw sequence data to identify different states or to distinguish body sites where that sample came from? And I just want to explain a little bit what is taxonomic analysis. This is not part of computer science or AI curriculum. So as you're aware, in biology, in the whole biological world, there is a huge taxonomy, but the microbiome taxonomy uses a hierarchical system to classify microorganisms. So these are very broad domains for specific species, and the levels include domain, which is the highest level, like bacteria, archaea, and eukarya. Then at the kingdom level, so microbiome studies, this is less emphasized. So because there is dominance of bacteria and archaea. Phylum, this is within a major division, and you will hear terms like firmicutes and bacteroidetes in the human gut. And then the next level is subdivision of a phylum, which is a class. For example, Clostridia in firmicutes. And then order is Clostridialis within Clostridia. And then family, which is related to genus or in plural genera, like Lacnospiraceae within Clostridialis. And then genus level, this is where mostly we're going to live in, these two levels. So Rosburgia within Lacnospiraceae, and then the most specific level here, groups of organisms that can interbreed, and here is a case, Rosburgia intestinalis within Rosburgia. Now, how does that look like data-wise? There will be these terms that we already mentioned that you will frequently see, which is amplicon sequence variant, as well as operational taxonomic units, ASVs and OTUs. If we relate that to this picture, ASVs would be the dots and OTUs will be the clusters. So ASV is a precisely resolved DNA sequence, which is inferred from amplicon data. And OTU is a cluster of DNA sequences. Typically, we cluster them, we're grouping them together based on similarity at 97% identity. So these OTUs help simplify the analysis by grouping closely related sequences. So with this, I think I can pass over to Amy. Amy, I didn't realize I took longer than anticipated time. I should have. So that we can walk over the notebook, the starter notebook, and look at what we are going to do here.
Great, thank you, Nevenka. Ray, do you want to pop in just for a moment to say hi and tell us why you're so excited about the challenge?
Yes, and apologies for being late. Right at the start, my boss's boss called me up, which I can't say no to, unfortunately, as is always the case, right? Yeah. So, yeah, but very excited about the challenge, and very excited to see all of you participating and interested in the challenge. For us, this is really an opportunity, to hopefully advance science and make some advances in this area. That's really what we're looking at from a Phillips point of view. We think this standard, this MPEG genomic standard, is a very powerful vehicle to help people doing science and research, but also even production work on genomics data. And we're hoping that the participants here, including all of you, will be able to take this, run with it, and come up with some interesting discoveries, which I'm sure you're going to find some interesting outputs from this. I also want to really thank Amy for managing all of this, and Jan for helping to lead this event, and Nevenka, of course, as well. So I'll stop right there so Amy can talk about the important thing, which is the notebook and how you can work with the data.
Great, thank you so much, Ray. Before we get into the notebook, we've had almost an information overload, which is quite a blessing. I'm going to pause, and if anyone has any questions for Nevenka, Jan, or Ray, please do raise your hand. We're just going to have a quick 5-minute Q&A, and then I'm going to get into the notebooks and some of the practicalities. Any questions so far? If you don't want to raise your hand, you're more than welcome to put it in the chat. Yes, Eddie.
Okay, so thank you so much, Ed, for the session timeout. Can you hear me, please?
Yes.
Okay, just my question is for Nevenka. For the longitudinal data study design, I want to ask, the samples of for stool, skin and oral, and also nasal collection of the sample, were they also collected within the 3 months of recruitment?
If I understood the question, so it's about the samples and
Yes,
the frequency at which they were collected?
Exactly, yeah. Would it still within the 3 months of recruitment?
Mm-hmm. Yeah, so the regular intervals were at 3 months, but if someone got an infection, say runny nose, or they were vaccinated, or they were taking antibiotics, then they increased the sampling because the expectation was that the microbiome will change, and that was an interesting event to sample because there is usually a stable or relatively stable microbiome over time. So that's why, and of course, it's costly to sample and to sequence. So that's why it's every 3 months.
Thank you so much. Thank you.
I just want to add on that at the beginning of the study, as Nevenka mentioned, there was a blood sample was taken with some extra information at the start of the study, with then those following nasal stool, those 4 body site samples that were taken every 3 months. Eric.
Hi, thank you very much, Emily, and also thank you, Nevenka, for the presentation. With regards to the papers that were within the presentation, I'm wondering if we could get them, because I don't see them on the funding sites. I don't know where we would find them if we don't get the videos to watch. Can we get the links to the references so you could read more.
Yes, so some of the slides have been updated, and I will add them to the competition, including Nevenka's really nice slides there. And then I just want to say that Nevenka, Jan, and Ray are on the competition discussion forum. So if you've got any questions that you haven't thought of yet, you're more than welcome to add them onto the discussion forum. Any last question before I jump onto the notebook? Brilliant. Let me just make sure that I'm on the right tab. Okay, so this is, I'm going to be looking a bit off to the side. I'm not ignoring you. I'm just reading through the notebook. So the first part of the notebook is to decompress the MPEG files. Jan showed us that there's what looks like two simple bash query scripts, which they are, to decompress and uncompress. Jan kindly decompressed these massive FOST-Q files into MPEG-G format, and so we're going to be extracting them. So first of all, we need to make sure that we've got Docker installed. On the competition page, there is how to install and how to start using Docker for Mac and Windows and Linux. So that's all on the competition page. Please note that it is near impossible to run Docker on Colab. So you will need to run this this notebook specifically on your local machine in your local Anaconda or Jupyter environment. So start one, make sure that you've got Docker, make sure that it runs. You can just do a quick hello world to ensure that it runs. Next, we need to import bio or biopython, depending on which library and where you're running your code. Just make sure that you've got bio or biopython installed. You do not need your machine learning libraries for this part of the notebook unless you're running the notebooks in quick succession. Then you can keep these as installed in your environment. But we do need bio, we do need our matplot, our numpy, our pandas, as well as our zip file and operating system. On the competition page, you'll note that there's a train files and a test files. These are two zip folders that contain the MPEG-G files. So first of all, we just extract them into our local directory. We ensure that we've got the latest version of Genie, which is quite a nice one-liner code. We then are going to look at one specific file from the test file. We're just going to see what it looks like, how to extract it before we go into bulk, just to ensure that our pipeline is working for one file, and if it works for one, it should work for all. So we're just going to be looking at this IDZZ. We are stripping off the last 4 parts just so we can ensure that we've got our train directory and our output FOST-Q. So this is just where everything needs to go, where we're getting it from, where it's going to, and what does our output file look like. This command over here is exactly what Jan showed us earlier on his slides. I've just broken it down into a couple more lines so that we can see where our container directory is, where our file name, which is IDZZ, goes to. And then we inspect our structure. You all know it's not working if it doesn't print out the whole Genie. It took me a while to figure that one out, but if you're not printing out the whole Genie and you get a standard output error, that means there's a problem, and please go back, ask on the discussion forum. This takes about 10 seconds per file, so this is quite fast to open up when you're just going slowly. We then get to see what does the FOST-Q file actually look like. So remember, this is a FOST-Q file unzipped or uncompressed. So in this one FOSS-Q file, we have got 160,000 different reads or different rows of lines of information. An average read is about 124.5, and the average quality score is 33.3. Jan explained this to me. The difference between a FOSS-Q and a FOSS-A file is it just doesn't have this quality score. So you can see if that's useful or even important in modeling. I just wanted to print out the first 3 reads just so we could have an idea of what that looks like. So we have a unique identifier for each read. And here we have our ATGC word or sentence. And this is where LLMs might get involved because we have a bunch of letters. So immediately we can start thinking about language. So keep that in the back of your mind. And is this a word? Is this a sentence? Is this even a whole book? And here we have our quality score for each read. There's a bit more information about the compression techniques, what is the output format, and then here comes the long part. I suggest making a cup of tea, getting a book, and running this piece of code, and it just shows you how powerful MPEG-G is, that it took me almost all night with multiple cups of tea to extract all these files. And that's just how big the FOSS-Q files are. And with MPEG-G, we now just get to almost zoom them across the world at a fraction of the price, which is significant, especially when you're talking about server costs, and a fraction of the time. So this is step 7. It's exactly what I've shown you over here. It just goes a lot faster. Remember when we printed out that really nice Genie statement that had Genie in cool text? I've commented it out here because there's a caution when running this, as it takes up memory just in your notebook, your Jupyter notebook. So don't print it out every time. And then I have just decided to go for all train, all test files. Let's go in one shot. I really recommend breaking them up into batches of maybe 500 or even 50 each, just to make sure your pipeline's working before going through. I'm going to pause for a moment and ask if anyone's got any questions on how to decompress an MPEG file. Welcome to raise your hand or put it in the comments. Great. Good job, guys. That means I did an excellent job at explaining, and we can go on to our starter notebook. I'm going to scroll up to the top. So again, we need to install bio. This is where we are getting counters and GC content, things that are bioinformatics terms, which we'll go into a bit later with Nevenka. Here we definitely need our modeling libraries. I've chosen to use a random forest classifier. It could be the best choice and it could be the absolute worst choice. This is where you get to explore. If you think you've got a better idea, please do try it out, go for it. This is a starter notebook and it's called a starter for a reason. I've given you a train, a test, and a train subjects file. So in our train file, we've just got our file name, so ID LETP. It's a stool sample. It's from subject B. And then really importantly, or just as a side note, it's from sample A. So when we look at cytokine data, you'll see that sample A has got a stool, a nasal, a skin, and an oral counterpart with it. So one sample talks about 4 different, one cytokine references 4 different samples. That took me a while to wrap my head around. When we're going through it, our train folder has got 66 participants in it. If you read the study, you will be able to determine how many participants are in test. We've got 4 unique sample types, which makes sense. which is stool, nasal, mouth, and skin. And we've got almost 3,000 samples. Again, if you read that sample, you can figure out how, read the paper, you'll be able to determine how many samples there are in total. This is just a distribution of number of files per user. So Eddie, this is to your question. Most subjects come in and they have about 25 or 20 samples each. So that means that they've come in more than once, because when you come in once, you get all 4 samples taken. We then have some incredibly passionate participants, such as this person over here, that I think almost swabbed himself once a month or once a week. And that just shows that this really is important work, that people are willing to come in almost weekly to get these samples done and to contribute to science. So thank you to participant number 450. We really appreciate you. Looking at the test files, this is where we were possibly evil. We only gave you the file, and this is your task. Can you determine who or where it's from? And then as a wink, wink, nudge, nudge, for the second challenge, can you determine who it's from? Are there any other interesting factors that you can find? We've told you a bit about the subject, such as their FPG class, which was taken from their blood test at the start of the project. If they were insulin resistant or insulin sensitive. For challenge number 2, are these something that you can predict, something that you can determine? And this is where Nevenka's causation or correlation comes in, which is something to ponder on. I do a bit of discovery on the subjects. Again, this is important for the second challenge. And here we get to explore those FOST-Q files, which has now taken up all the memory on your computer. And this is, again, MPEG-G wins. We do some statistics. We count the number of ATGCs. I sillily decided to rerun this notebook today. And so that's why it has stopped outputting over here, just because the FOST-Q files are so large. But please don't be intimidated. I've done this before. I've ensured this notebook runs and it does work. So these are some of the stats you get. When you look at the GC counter, you'll see that there's a nice Gaussian distribution. And that just confirms what we are looking for and what the paper also confirms. Okay, modeling. As a non-bioinformatician, I just decided to look at the number of reads, the average read length, the GC content, and just the number of, or the ratio of ATGCs in either reads. So these are all the features I was using, but Nevenka came up with some brilliant ideas that she's just going to comment on, and these will hopefully prompt you and make your brain go. Over to you, Nevenka.
Yeah, so thank you, Amy. I think this is where your brilliance needs to come in, but from bioinformatics point of view, and I have a paper on sequence modeling from, oh boy, now it's 2 decades ago. You can extract dinucleotide count, trinucleotide count, K-nucleotide count, just to create features and to see what is present in the data. That's simple to think through, but I think what would be even more fascinating and challenging would be to combine LLM technology with this idea of how to model gene and microbial information, which is encoded in these sequences with A, C, Ts, and Gs, and how to essentially treat DNA as a language. And now a gene can be, or a gene sequence can be considered a sentence. And then parts of the gene that are that have different role, different exons, those can be words. So here is where you need to dive a little bit in biology, or maybe your LLM will be able to figure out how to tokenize and what tokens should be. This is definitely an area where we are looking for some ingenuity. There is a model out there, it's called EVO, it's by the Arc Institute at Stanford. You might want to look at the paper. They have developed this large foundational model, and maybe you can use that model. Nothing is off limits. You can get more data from other studies that I mentioned, but also look at what else has been done in this space. And if you know how to stand on the shoulders of giants, please go ahead and do so. This is what I think would be the most exciting part. Thank
Oh,
you.
Nevenka, one of my favorite quotes. Absolute favorite, and that's where I often sit, so thank you. So we just make sure that we've got all these features for all our FOST-Q files that we derived from our MPEG-G files. We ensure that we've encoded all our strings or label encoded all our strings. We do our normal, this is where normal machine learning starts, which we can all go, phew, we figured out the bio stuff. Now we can get into something that we're comfortable with. So we're going to define our features and targets. We're going to split them into a train and test ourselves. Remember that Zindi has a separate test. So we're just splitting it up into our X, Y. Now, here we do our train test split for evaluation. We run our random forest classifier. This model, again, is called a starter notebook because I'm only using 10 estimators, which means we're not getting a very good score, and there's lots of place for you to improve on. I've also only chosen one model. What happens if there's multiple models? What happens if an ensemble is going to do something interesting? So those are things to keep in mind. We then evaluate and we get our classification report. Oops, my notebook broke while running through it, but I promise you it's there and it's quite exciting. What we did with our train files, we need to do the exact same on our test files. So we need to extract those features, which was that function we built higher up. And we get to predict it onto the test set. Finally, we just need to ensure that our column orders are correct. Please note the column order. I'm going to say that again. Note the column order before submitting. And finally, before we make our submission, we need to set our index to false. So what we've just done, we've created this brilliant centralized model. Oh my gosh, we're super proud of ourselves, and you should be proud of yourselves. It's a massive feat. But that's not where the challenge ends. We need to take it one step further. We need to start looking at federated learning. Federated learning is really important, especially when we could have some unlabeled samples, some samples where features don't overlap with each other. It's also incredibly important in the medical domain where we don't want to be sharing data with multiple people. So if you're in the top 10 of the private leaderboard, once you've done your submission selection, you will need to have your federated learning solution at the tip of your fingers to submit immediately, or you've got 3 days to prepare it. So to submit your federated learning. So we ask you to do 3 big leaps. First, get those FOST-Q files from MPEG-G and understand the power of it. Figure out, how do LLMs fit in here? How does normal machine learning, how does deep learning help with classifying samples? And then finally, what is the importance of federated learning? And how can you turn your beautiful centralized model into a federated learning approach? I hope that you could have all seen my screen as I went through, because it's just popped up on mine, but no one said anything in the chats, which is great. I'm going to, oh, thank you, Eric. Phew. I'm going to pause and ask if there's any questions or anything anyone wants me to go over. Again, I did a brilliant job, guys. Thank you. Just a reminder that this recording will be made available tomorrow and at latest Monday, so you can come back. If you're stuck, we're all on the discussion board, all willing to help, and there's some real giants there that you can ask for help. Awesome. Okay, thank you, everyone.
Thank you, everyone. Thank you, Amy. Thank you, Ray.
Thanks all.
Bye.
Thank you.